So, hello everybody. It is my pleasure to present another seminar, this time from the European Astrobiology Institute. It is given today by Kristina Beblo-Vranesevic from the German Aerospace Center. Like me, Kristina did her PhD at a university located at the banks of the river Danube. In my case, it was Vienna, and in her case, it was Regensburg. But during this time, she already worked at the German Aerospace Center, where she is now a scientific employee. And she is especially working on the EU-funded project MASE, Mars Analogues for Space Exploration. Welcome, Kristina, and I'm looking forward to listening to your talk. Many thanks for coming to all of you.
Thanks, Wolf, for the kind introduction. Good afternoon, ladies and gentlemen. Hello to everybody. My name is Kristina Beblo-Vranesevic. As Wolf already introduced, I'm from the German Aerospace Center. But first, since 2 years we are working online, and I'm still sometimes confused. So I stopped at the word German Aerospace Center. So I'm from the German Aerospace Center in Cologne, where the headquarter is. And the building where I'm working is building 24. It's split into 2 buildings. It's here and here. And at our institute, the Institute for Aerospace Medicine, we are in the Department of Radiation Biology, and I'm working in the working group Astrobiology, which is led by Petra Redberg since many years. And we have different labs, and we have some space simulation facilities in our ground facilities. So our ground facilities to simulate different aspects of space. But now we are switching our view. So if we are thinking about Mars today, this is a typical view from Jezero crater. And the main question is, what do we have to consider if we want to know, is there or was there life on Mars? And the first point we have to consider is the atmosphere on Mars today. On Mars, there is a very low pressure. It's much lower than on Earth. We have anoxic conditions. We have only 0.14% oxygen, and we have very low temperatures, minus 65 degrees on average. Additional to these atmospheric conditions, we have the Martian regolith itself. So the Mars is covered with soil, with regolith. It's very dusty. In this regolith, we find, or several missions found, oxidizing compounds like perchlorates or hydrogen peroxide. We have there in some areas high salt concentrations. There are very low amounts of nutrients, and there is no liquid water flowing around. It is very drought. Additionally, we have higher influx of radiation on the surface of Mars. Mars do not possess an ozone layer, so solar UV radiation in the range of 200 to 400 nanometers and even lower are hitting the surface. Mars has no magnetic field, so also cosmic ionizing radiation is hitting the surface. And taking this all into account, the surface of Mars is a very deadly and even poisonous environment from the point of view from humans. So the big question of the MASE project, Wolf already told you, was, are anaerobic microorganisms from extreme terrestrial environments, which are somehow similar to Mars, so we are talking here about Mars analog environments, are these organisms more tolerant to Mars relevant stress factors? And so In 2014, MASE was started. It was a EU-funded project for 4 years, and it was the first time that we take a close look to microorganisms and analog environments which were anoxic, because on Mars we will not see that much oxygen as we have on Earth. And in MASE, we thought it's a good idea to collect new samples from these anoxic environments, isolate microorganisms, and deposit them in culture collections to make them available for other researchers. We wanted to investigate the limits of anaerobic life and the tolerance of these microorganisms to different Mars relevant stress factors. This was the part where DLR came in. Additionally, in MASE, we wanted to study the preservation potential of different organisms. Of these organisms, we isolated from these anoxic environments, and we wanted to test some live detection equipment. So the first step was to define in Europe different Mars analog sites, and we came up with 5 different sites. We had the Rio Tinto, we had a cold sulfidic spring nearby Regensburg in the Sippenauer Moor, we had a lake in Iceland, Lake Grenavatten, we had a potash mine, the Bowlby salt mine in the UK, and we got some permafrost samples from Iceland. And we went to these Mars analog sites and took samples. Back in the lab, we did some inoculations. So we had more than 1,000 different conditions of our samples. We had hundreds of enrichments, and finally we got 31 facultative anaerobic isolates, which were stable growing. And these are these 31 facultative anaerobic isolates. And it turned out that especially 2 organisms were very interesting because the growth was very reliable. And with these organisms, we worked further on. And we stressed these organisms. We wanted to know if Buttiauxella and Yersinia are able to survive desiccation, radiation, vacuum, different temperatures, the exposure to oxidizing compounds, and the lack of nutrients. But in recent times, so since 3 or 4 years, we added another Mars analog site to our list, and these are deep sea brine pools. And what are these deep sea brine pools? These are large areas of brines on the bottom of the ocean, and they are really separate from the water over the pool, and you can really dive into these brine pools. And every brine pool has a unique combination of physical and chemical parameters. For example, the temperature, the pH is very different. We have different minerals and a different chemical composition. And from one brine pool here in the Red Sea, from the Shaban Deep, we got an organism. It was isolated several years ago, 2003, by André Antones, and it's called Salinisphera shabanensis because it's coming from the Shaban Deep. And with these model organisms, we worked further on. So in the next half hour, I will talk mainly about 3 main organisms. It's Buttiauxella, MASE EM9, Salinisphera shabanensis, and Yersinia intermedia. All these 3 organisms are bacteria, all are mesophilic organisms, and all are facultative anaerobic organisms and coming from different Mars analog sites. Coming back to our first view on Mars today, we thought the main focus should be on desiccation because we will not have liquid water on Mars. We have the radiation, we have the perchlorates, and we have the soil. On all these parameters were tested in our labs. So how is it done, the general workflow? So first of all, we have to cultivate our organisms. Then we desiccate them. This is mainly done on glass slides under anoxic conditions. We can then expose them to vacuum or to Martian atmosphere. We can irradiate them in liquid or after desiccation. And then we determine the survival rate by plating, and we do some microscopy to see if the cells are somehow changing due to the stresses. And here are the first results from Butioxella, Salinisfera, and Yersinia about desiccation and radiation. What we can see, we tested the desiccation up to 200 days under anoxic conditions, and we see that all three behave different. The same is the case when we are exposing them to ionizing radiation with doses up to 3000 gray. This is sounding not too much, but please keep in mind, for example, humans are really dead after exposure to 10 grays. So this is still a lot. And this was not only visible in the ionizing radiation, we saw differences also when we irradiated the cells with UVC alone and monochromatically and with polychromatic light. Polychromatic light means these are the wavelengths which are hitting the surface of Mars. So we have a Mars simulation lamp where we can irradiate the cells like it would be on the surface of Mars. And if we compare the F10 or F10 values and D10 values, which means that after this irradiation, the numbers of survivors is reduced by 90%. We see that it's quite in the range as other model organisms like Escherichia coli, and but it's lower than the model organism in astrobiology and in radiation biology, Deinococcus. Our model organisms do not survive as good as Deinococcus. However, coming back to desiccation and radiation alone, it was proposed several years ago that both resistances are combined. So it was proposed on the data from Deinococcus that all organisms which are able to survive desiccation are also able to survive radiation. And this was one point we at the early beginning of the project, we saw in our model organisms, we do not have this correlation, which was proposed several years ago with Deinococcus. The next step was to combine the desiccation and the ionizing radiation. And what we saw is there is an additive effect. So if the cells are desiccated and irradiated, the survival rate is lowered, as you can see by indicated by the blue arrows. However, for Yersinia, we saw that the dried cells were more tolerant to the exposure of ionizing radiation as the cells in liquid. This was not the case for Butioxella. Up to now, we do not know the exact reason why this happens, so we can still speculate, but maybe the cells are forming somehow crusts during the desiccation and can repair afterwards the damages from radiation better than in liquid. In general, ionizing radiation will only play a bigger role over geological timescales on Mars. So even on Mars without a magnetic field, we do not have that much ionizing radiation on the surface as we use here in our experiments. Coming back to desiccation, the question was, if, for example, organisms are on the surface of Mars, they are in contact with the Mars soil, with the Martian regolith. And what happens to desiccation, to the survival after desiccation, if they are in contact with this material? There is a company in the US, Exolith, and there you can order Mars Global Simulant. It's also called Martian regolith, and you can buy it in kilograms. And we added additionally sucrose, which is known that sucrose can help cells during desiccation. And what we saw was very impressive. So these columns is the survival of Butteoxella and Salinisfera if the cells were mixed with the MASE soil and the sugar. And we saw that the survival is much higher than if the cells were dried without Martian soil and sugar. So the desiccation tolerance in general can be influenced, and we have a huge protection potential due to the added Martian analog material and the sucrose. The question was, why does this happen? So this was never shown before, that Martian material can help cells. And we had a closer look with electron microscopy. And if we look very closely, we see in this Martian soil this hair curl-like structure. And we know that this is Kieselgur or Diamoschus ursus. And then we thought maybe the cells can bind to these structures, or these structures can help the cells. So we mixed the cells or grown the cells in the presence of the soil. We did some DAPI staining. DAPI is staining the DNA of the cells. And we saw, first of all, the DAPI is not staining the material. As you can see here are the different particles of the soil. Here you can see these hair curl like structures, and we saw that these are not stained with DAPI, but we saw that the cells are somehow attached to different grains, and they are growing in kind of clumps. So we are still not sure if they are really sitting on the grains or if they like to attach to the grains. And we are still working on the question how the cells are connected to the grains of the soil, but probably this agglomeration effect is the answer why the desiccation tolerance changed in the presence of the Martian soil. The next point was, if we are thinking on Mars, probably there will not be one Butteoxella or one Salinisfera on the surface. We tried to mix the cells before the desiccation, and we wanted to know if they can help each other or if they do not like them, the other species. And we saw that if Butteoxella and Salinisfera is mixed before desiccation, the survival is slightly better than separately. And we worked further on, and we did a desiccation experiment together with Deinococcus geothermalis, also very desiccation tolerant organisms. And we saw that in this artificial community, the survival is better than if the cells are alone. Another form of desiccation is the exposure to vacuum. First of all, we have to dry the cells and then expose them to vacuum, and we can also expose them to Martian atmosphere. So we did this with Yersinia, and we saw an influence of the pressure. So this is the influence of the vacuum, and we saw an additional influence of the Martian atmosphere. So both conditions are also additive and reduce the survival rate after desiccation additionally. Then we tried the combination of polychromatic UV in liquid and the exposure to polychromatic UV after desiccation, also in the presence of the Martian soil. And we saw that there's still survival after the combination of both stresses. So here we have 12,000 joule per square meter of polychromatic UV. And we saw that also here we have a slight positive influence of the Martian soil on the survival if the desiccation and radiation is combined. Another important point on Mars, I already said this, are the perchlorates. So in former missions, perchlorate ions were detected on the surface on Mars. We have their concentrations between 0.4 to 0.6%. And on different locations, we have different perchlorates. So we have sodium perchlorate, calcium perchlorate, and magnesium perchlorate. And we were thinking, what happens to the cells if they are exposed to different concentration of perchlorates as a single stress? What is happening if the cells are growing in the presence of sodium perchlorate? And what happens to the cell if they are treated with the perchlorates and then if they are desiccated or irradiated? Here is the exposure experiment with sodium perchlorate. Just to let you know, these blue lines are the concentrations we have on Mars. So these are the 0.5 and the 1% sodium perchlorate. And we tried this with much higher concentrations for 15 minutes. And we saw here that Buttiauxella and Yersinia are in the same range as other standard model organisms, Escherichia coli, Deinococcus, or Bacillus subtilis as vegetative cells. Here are the results if we store the cells in the presence of the Martian concentrations of sodium perchlorate. And the black and light gray ones is Buttiauxella and Yersinia intermedia. And we saw that the storage at room temperature without oxygen of the Martian concentrations of sodium perchlorate did not change the survival rate drastically. However, the big question was, what is happening if the cells are growing in the presence of sodium perchlorate? So please look especially here for Buttiauxella and Yersinia. This is are the cells under standard conditions, so we have short rods. And if they were growing in the presence of perchlorates, we see that the morphology of the cells is changing. We get these chains of cells, which is a reaction to the stress. And we saw this not only in these different organisms we isolated from Mars analog sites, we could show this also for organisms from hydrothermal system, Hydrogenothermus marinus. The next step was a combination of perchlorates and desiccation. To make this long story short, perchlorates have species-specific effects. So we saw that the reaction of the cells to the different perchlorates is different. They are somehow similar in their influence, and mostly 1% of the perchlorate is more influencing than the 0.5%. The picture is a little bit different with the combination of radiation. Here the results for Yersinia are shown, and here the perchlorates have no influence on the tolerance to ionizing radiation. The blue line is without any perchlorates, so all survival lines bundled together, and there is not a really significant difference. There is a little bit of difference if Buttiauxella was first cultivated in the presence of perchlorates. So Buttiauxella is making these chains, and if these chains are irradiated, they are a little bit more tolerant to UV radiation and to ionizing radiation. So here we have the difference to the treated cells and the not treated cells. With Buttiauxella and Salinisfera, we did another big experiment in cooperation with NASA. We did a stratospheric balloon flight Also, the stratosphere can be seen as a MASE analogue. So if you compare the conditions during the balloon flight and the conditions on the surface of Mars, we see that it's somehow very similar. Therefore, we mounted this small box I already showed to you, this T-Rex box, on this gondola of the balloon. And in this box, we had Salinisfera and Butyaoxella. Then the whole mission, the duration of the whole mission was 5 months. So the cells were dried for 5 months. And the stratospheric flight was for 5 hours in 38 kilometers height. And we saw that Salinisfera survived this whole treatment, which was not only which we could not only see in the survival rates, also in an experiment to determine the metabolism of the cells after the treatment. We could show that the cells are still alive, but we could not show any survival for Butyaoxella. It's important to say here the cells were in a naked form. There was no shielding material or Mars soil. So we had only the cells, and probably Butyaoxella just not survived the 5 months of desiccation. So to sum up the results I already showed, in the MASE project, we were able to simulate selected environmental factors of the Martian surface, as I already said, especially the drought, the radiation, oxidizing compounds, and of course the anoxic conditions. And we could show that the new model organisms from Mars analog sites are able to survive some of these factors. So the desiccation and radiation tolerance is not correlated. Desiccation partly leads to enhanced radiation tolerance, even we do not know why. Martian relevant concentration of perchlorates during the growth lead to cell changes in cell morphology. And perchlorates have an influence on the desiccation tolerance, but not on the radiation tolerance. And normally here the question is coming, Kristina, do you think that there is life on Mars, or is life on Mars possible? And I would say on the surface, we have different conditions, and all conditions alone, cells are able to survive then. But the complete package of all conditions in parallel, these are also for microorganisms very challenging. The situation is changing if we go a little bit deeper. So if the cells are protected from the radiation on the surface, and maybe there is some salty brine solution in the deeper areas, the probability that there could be life is much higher than on the surface. But in general, what is the next step? The next step is to use our new model organisms for a space experiment. And we applied 2014 for a space experiment called MAXEM. It's Mars Exposed Extremophiles Mixture. And MAXEM will be a passive exposure experiment on the outside of the ISS. And in MAXEM, we will not only use anaerobic microbial communities on isolates from these analog sites, these new model organisms. We will also use other archaea and bacteria, but also higher organisms like ciliates and viruses. Additionally, we will have artificial fossilized microorganisms to investigate further if they can be used for big biosignature identification. The total mission duration of MAXEM will be around 6 months. The exposure time in space on the outside of the ISS will be about 3 months. We will have the Mars-like UV spectrum. The launch is planned in 2024, 2025. It's still not finally fixed, but we are still crossing our fingers that MAXEM will fly within the next years. And we will have a ground reference in our simulation facilities, a parallel experiment, which is really mandatory for the analysis later on. So these different MAXEM samples are grouped into 6. We have these group 1, the original samples. We have group 2. These are isolates which are protected. Probably we will use the Mars soil as a protection material. We will have in group 3 the pure single cells. Group 4 are defined mixtures or artificial communities, which I already shown in the experiment with Deinococcus, Butteauxella, and Salinisfera together. We will have some strains from the ISS. So there was an experiment called extremophiles where new organisms from the ISS inside were isolated, and these will go back to the ISS in MAXEM. And we will have in group 6 the fossilized cells. Even we are working since several years on MAXEM, we have still not finally fixed which analysis we will do after the cells are coming back. Of course, we will have some cultivation-based methods and some cultivation-independent methods. We will look for the survival, for the metabolism. We will look for the morphology if there is something changed. But also we will use different, probably also some omics techniques. We will look if the cells are producing different proteins, and we will look into the DNA integrity. But as I already said, this is not finally fixed, and this is the only advantage of the delay. We have still time to test and to think what we want to do after the return of the cells. And with this, I'm at the end of my presentation. I thank you a lot for your interest. And now I'm open for questions. Either you ask me directly, or you can ask me in the chat, or if you think about the presentation later on, you can write me, of course, an email.
I have a question regarding the ionizing radiation that Mars surface is exposed to. All of your experiments seem to have been done by radiation sources whose wavelength goes from 200 nanometers to 400 nanometers. How do you account for the galactic cosmic rays that are incident on Mars, especially because there is no magnetic field on Mars?
Yeah.
Thank you.
First of all, you have to divide in this ionizing radiation, this galactic cosmic radiation you mentioned. This we cannot simulate these galactic cosmic rays in our institute or in our labs. So we use an X-ray machine to simulate the ionizing radiation. And on the other hand, you have this non-ionizing radiation, this UV-related radiation between 200 and 400 nanometers. There you have lamps which are emitting these wavelengths, but it's not possible to have lamps with lower UV ranges. And that's the reason why we want to go to space, because in space, of course, you get exactly these radiation which are not that easy to simulate on the ground.
Thank you very much, Kristina. I appreciate it.
And I can read the second question in the chat. Did you ever look to the resistance of yeast in these extreme conditions? Yes and no. So we have currently a PhD student who is working with yeasts, and but he is mainly interested in extreme conditions more related to Europa and Enceladus. So we have other extreme conditions. These Martian extreme conditions I never did these experiments with yeasts, but however, this could be an option in the future that we add another new model organism from the yeast, or which would be also very interesting, to add model organisms from the archaea to compare if the different domains of the tree of life behave in the same way as the bacteria did. There is a question, what do you think about experiments directly on the Mars? Could it be possible in the future? So the problem is that we can go to Mars, and at the moment we have no techniques to come back with samples. So the complete Mars sample return mission scenario is still not completely thought. So it's not possible to bring at the moment samples back. So we have to do all our experiments, would have to do our experiments remotely on Mars. From my personal opinion, it would be super interesting to do experiments there, but I think from a technical point of view, it will be very, very difficult to go there and to analyze there, get the results back, and to analyze the results which were made on Mars. I think and I hope that in my scientific career, I will be able to work with samples from Mars. So the Mars sample return is planned to be within the next 20 years. And also there, fingers are crossed that someday I get a little piece of Mars to see it in real.
Hi, I would have first of all, I've also heard that due to microgravity, for example, cancer cells do clump under microgravity conditions. Could this, in combination with sodium perchlorates, intensify their radiation tolerance?
In general, this clumping can be an answer of the cells due to stress. So here we stress the cells with perchlorates and they do the clamping. This can also happen in microgravity. So the cells are stressed maybe due to microgravity and they are clumping. What we see, if the cells are growing in these clumps, they are more tolerant. So they are more tolerant to desiccation, they are more tolerant to radiation. The complete answer why this is happening, we do not know at the moment. Probably there are some shielding effects. So if you have a clump and the irradiation is coming from one side, maybe the outer shell of the clump is dead, but the inner cells are still protected. Maybe it's a kind of small biofilm that the outer cells are a protective layer for the inner cells. And this can be true, of course, for the radiation. We are still not convinced that this is the answer for desiccation, because if the water is gone, the water is also gone inside. So they cannot hold the water in their clump. So this is cannot be finally answered, but of course, I think this clumping or agglomeration, or how you will call it, can lead to enhanced tolerance to different stresses, not only to perchlorates, to radiation, to desiccation. And this maybe can be an option that the cells are growing in these clumps and then survive probably on the surface of Mars better than as single cells.
Okay, thanks. And second question, because I'm a huge fan of archaea, why always choosing bacteria and not archaea?
One of the very surprising result of MASE was in these 31 pure cultures, we got 1 archaea and 30 bacteria. So the MASE project was to work further on with these organisms. In the past, I worked a lot with archaea, so I did my complete PhD with more or less archaea. And we saw that also there are desiccation and radiation tolerant organisms also there. This correlation does not exist between desiccation and radiation tolerance. However, the big problem for us at the moment with archaea is that the archaea, we were mainly working with hyperthermophilic archaea, and it's not that easy to grow them on plates. And without plating, the whole experiment got a lot, it's much more work to do it with dilution series to get to the step, to get a survival curve. And at the moment, it's not that doable that we do it without plating. It's a pity because I think probably since archaea are much older than bacteria and more robust, and you find them in extreme environments, you find them under condition where normally nothing should grow. These would be the perfect model organisms if they would grow a little bit more bacteria-like.
Okay.
But I think if you're working with archaea, you know this.
Hey, thanks a lot. An awesome talk, honestly.
Okay, I have a
some
question.
further questions? Yes, Victor, please.
Yeah. Thank you, Kristina, for your nice talk. If I don't remember, if I remember well from your slides about the radiation experiment, the doses you are using is at the level of few grades of even hundred of grades. The point is, as the Hasler et al., I don't remember, 2014, something like that, reported that at the Gale crater, the radiation was around 0.2 milligrays per day. I wonder whether this, in fact, this radiation is relatively low for I'll bet that those microbes you have shown us will perfectly grow under this condition. It shouldn't be a true problem. But what do you think about that? Do you think this radiation is I don't know, it can be somehow low and microbes can survive?
So you are completely right. The dose on the surface of Mars per day is really, really low. So our microorganisms will not care about it. But they will care about it over geological timescales. So if you're thinking maybe there was life on Mars and they are hiding somewhere, maybe they are encrusted, like you see it also in salt mines, where in salt crystals cells are trapped and lying there for millions of years, then of course the ionizing radiation can accumulate until higher doses.
Yeah, but excuse me, sorry to interrupt you. This is the case. We are assuming that life under this condition is not having any metabolic activity, no repair, no nothing. Okay, but just in the case we have some humidity, some some. I mean, this radiation level, it shouldn't be. Should be something perfectly. That can be understood by life.
Yeah, so the ionizing radiation will not the big factor. The big factor on Mars will the UV radiation.
Yeah.
And so this is the reason why we more are going into the UV direction, because we think our organisms are surviving such high doses of ionizing radiation. This will not play a major role in our experiments. That's completely right.
Mm-hmm. And just another question: in your experiment with the perchlorate, these changes in cell morphology, have you associated to any? I don't know. Have you seen any growth, or it just I don't know, it's associated with some kind of growth, or just because a cell deformation? Anything you can tell us about this?
So we saw that if the cells were growing in the presence of this perchlorate, they are building these strange structures. So they are not able to divide anymore. Probably the perchlorate is influencing the division of the cells. or the cells remain together to help each other. So it is known that also if the cells are still connected, they can exchange particles. So maybe they help each other or they are not able to divide. But the complete reason why this, so we see that they are doing this, but we do not know the exact reason in the cells. So we do not know which gene is affected by the perchlorate. This has to be done in the future.
Thank you.
More questions, please? Eva.
So, may I? Thank you. Thanks, Kristina, for a very interesting talk. I would like to know what kind of conditions you were able to monitor during the whole flight, and how, I mean, important is to have the very good or precise measurements of different physical conditions you've got during the flight, because they are changing all the time. So can you comment on that?
We had on this, so it's a series of balloon flights done by NASA. So they do this in regularly at regularly times. And in our flight, we measured the temperature. We had 2 UV sensors. I think one was for UVB exclusively, and the other was UVA and UVB combined, I think, but I'm not completely sure. And our box was not directly monitored, so we had this completely sealed box delivered, and in the box we had our dried cells, and in the box we had the Martian atmosphere and we had the Martian pressure. So we call it we had this box under Martian conditions, and on the outside of the gondola we had the temperature and the UV sensors. And I think for some numbers also given in the publication, there were some calculations from previous flights, and they were compared to our flight in, because of course we had also the height of the balloon and the duration, and then it was assumed how much irradiation we got on our samples.
Great. And I mean, in analyzing your results, I mean, what was the most important? I mean, did you take this into account, or this was just for monitoring? There was no influence on the sample.
Of course, it is important because we had the UV exposed layer and we had a dark layer under the cell, under this UV exposed layer, and there were differences. So the cells which were on the top were much more damaged than the cells on the dark. And this is the influence of the UV. And of course, we have to take this into account that there was UV. And it's interesting that the balloon flight itself was only 5 hours, and we already saw an influence of the UV due to these 5 hours, and the complete mission was 5 months. So it was a very, very short time of exposure if you're taking the complete mission, and even this short exposure is leading to a change in survival. So it's super important to take the UV into account. And we had no shielding glasses or filters, so we used only quartz glass on top of our box. And this means if we want to expose cells to space, we probably need to use filters and reduce the incoming UV, because we do not want that all cells are dead. We want still some living cells back and want to analyze them.
Sure, thanks a lot.